Yeah, ladies and gentlemen, welcome and apologies. These slides are not yet in the lecture notes, so I will upload them afterwards. And with high probability there will be little of that lecture in the exam. So it's not that serious if you don't have the lecture notes, but I think it's an exciting topic which we should have in mind, and therefore I assembled a very personal view from an engineering side on optogenetics. So I like to introduce that from the more neuroscientific or biological side. So what are we talking about? And then I'll switch my hat and look from the engineering side. What does that mean? So what should we do? And I found my niche in, let's say, translational neural engineering. So the question, how can we make things robust and reliable? That's a bit different from the question, how exciting is that, and can we record something over a day or something like that, or a week? So my question always is, if we like to go the road down towards very chronic implantation time frames, would that still work, or should we consider something? And I like to walk you through that whole topic, starting with a video from Nature in the year 2010. And it started on that particular, here we go.
Scientists can switch on cells in this mouse's brain simply by switching on a light. The light activates nerve cells which make the animal walk in circles. This neat trick is also a powerful new tool. Using light to control the behavior of cells is teaching us about everything from how we wake up to how we learn. This is optogenetics. It all started with algae. In 2002, researchers discovered that a protein that causes green algae to swim towards or away from light is a light-sensitive channel. Blue light causes the channel to open, and positive ions then flood into the cell, much like the influx of ions that cause nerve cells to fire. Neuroscientists were quick to see the potential of these algal proteins. They realized that if they could get proteins called channel rhodopsins to work in mammalian nerve cells, they'd have a very precise way of controlling nerve activity in the brain. For many applications, this method would be better than drugs, which act too slowly, and better than electrical stimulation of the brain, which is less precise. Getting an algal protein into, for example, a mouse brain requires a bit of genetic engineering. First, you take the gene that encodes the protein, plus another piece of DNA called a promoter, and you put them in a virus. When the virus is injected into the mouse's brain, it infects neurons and delivers the gene. A chosen subset of those neurons will have the right machinery to activate the promoter, and in those cells, the algal protein is expressed in the cell membrane. To activate the protein, all you need is light, delivered via a fiber optic cable. It's the ability to target specific neurons in living, moving animals that makes this technique so powerful. And channel rhodopsins are not the only proteins in the optogenetics toolkit. In this fly, a different light-sensitive system is used to target just 2 out of 200,000 neurons. These neurons govern an escape response, the reflex that makes the fly flee as you move in to swat it. Here, a flash of light activates the neurons, making the fly jump and spread its wings. It can't take off because it's trapped in a petri dish. As well as activating neurons with light, scientists wanted to be able to silence them. Switching neurons off is just a case of using a different light-sensitive protein, such as halorhodopsin, and a different colour of light. Watch how this worm's workout is interrupted when a yellow light is switched on. The light activates halorhodopsin, triggering a flood of negative ions which inhibit the worm's motor neurons and stop it swimming. And neuroscientists aren't the only ones using optogenetics. As well as turning neurons on or off, a diversity of tools allow researchers to control many different processes in living animals and in cells in dishes. These mouse heart cells have been engineered to beat in time to pulses of light. And here, a light-sensitive protein makes this skin cell move towards a laser. The applications are endless. Researchers have already used light to transform the stumbling of rats with Parkinson's disease into a steady walk and show which nerve cells fire when a sleeping mouse wakes. Optogenetics is shining light on everything from animal behaviour to the interactions of proteins inside cells. That's why Nature Methods has selected optogenetics as its Method of the Year 2010.
So now we know everything and could go home, right? I've seen your faces, and it's a very enthusiastic, fundamental research biology view of the world, right? And some of you might have focused on certain aspects, and I don't want to comment it now, but I walk you through certain aspects that I think we should consider. No matter if we tend to work in neuroscience or in neural engineering later on with that technology. So to cut that long story short, we have a genetical modification of electrically active cells. That means light-sensitive proteins are generated in the cell membrane. We have then specific cell manipulation using visible light, and that was that promoter gene. So a kind of keyhole mechanism that we genetically, or not we, but somebody else, genetically engineers proteins with that famous code for those optical active channels that know where to go and where to generate those channels in particular cells. And the promise is high spatial and temporal acuity. If we would take electrical stimulation, the electrical stimulation does not differentiate. Is this a yellow cell, a green cell, a red cell? So talking in colors, that means it does not specify if this is a motor cell, a sensory cell, an interneuron, or whatever. Just takes whatever is in the electrical field and excites that. So that seems to be a very exciting approach to interrogate the brain and to find out certain aspects behind neurobiological questions. And this has been the method of the year in 2010. It developed tremendously since then. And there's a plethora of opsins and experimental paradigms, as we will see shortly. So it's not only now that blue and yellow stuff for excitation and inhibition, but we would have nearly the whole visible light now, and we have a variation in both the temporal response. So how long should I shine that light on and what does that do? And how much energy do I need? And when I discuss that intensity thing with my colleagues from biology, I lose my last hairs. That is really because the question is, when we talk about energy, what are we talking about? We'll find out here, that's milliwatt per square millimeters. That's not the energy, that's an energy density, first of all. So if we talk about 14 milliwatts, Is this 14 milliwatts or is this 14 milliwatts per square millimeter? And what diameter of optical fiber did you take? If you don't know what a square millimeter is or how large your thing was. And where do you measure that energy density? Is that on the tip of the waveguide? Is that what your laser spits out? Is it in between? Or so there are a lot of questions, and if you don't sort them out properly, that happened to us, therefore I know what I'm talking about, you come with something and it doesn't work at all, and then people might get angry and say, you told me that works forever, and then you said, yeah, you told me this is that energy density. We try to, you misunderstood that, we just meant per square micrometer, and then you recalculate that and you come up at the level that's 20 times higher than expected or something like that. So it's very important to learn that common language there, even if you believe that the other side is applying already that technology, does not mean that you talk the same language there. So that's one of the important issues. And if we now look more into detail, we have a brain, we have a nerve cell, we have those ion channels, and the idea is to stimulate with light and to record the neural activity. And that is another beauty. If you stimulate electrically and you want to record at the same time, those of you who have ever worked with an electrical amplifier know if you stimulate with high amplitudes and you try to record, let's say, immediately, you drive your amplifier into saturation, and then you have to wait for some seconds until that amplifier goes back to a working range where you can record something. And in these cases, quite often, your desired signal that you're looking for is hidden in the electrical artifact. Here, we have the idea, that's beautiful. We stimulate with light, we record electrically, nothing, no interference at all. That's only half of the truth. If you go a little bit more in detail into physical phenomena, you will find out something like photoelectricity. And that is not only something that works in semiconductors. You know that very well from photovoltaic cells. You put them into the sun and you get electricity out of that. That means the sunlight is transferred into voltage. A laser is at least as powerful as our sunlight. And if you shoot with a laser on a piece of silicon or another semiconductor, you generate charge. And your silicon doesn't mind if it's in the brain or on the roof. So if silicon sees energy, it generates charge carriers. That means if you don't take care with silicon probes in the brain, you might have that effect. On the other hand, the photoelectric effect works very well on metals. If you shoot light on metals, there is also change in conductivity by the photoelectric effect, and you might see artifacts there. They are much smaller than the electrical ones during electrical stimulation, but you might see artifacts. And this is something from the engineering side we should sort out when generating those probes, exactly knowing what kind of artifacts do we get, how long do they last, and can we suppress them, or can we at least teach the applicants to ignore them. So, this idea of recording electrical signals can be either integrated into silicon or into polymer-based electrode arrays, and that's a new playground for engineers now, trying to put electrical and optical modalities together here. And to do that, one could imagine, for example, to integrate waveguides. So we have seen those mice with their waveguides made out of glass and all the blue light that looked a little bit spooky there. So the question, can we integrate waveguides? Or should we integrate other stuff? Advantage or advantage and disadvantage, you have the light source outside the body. Disadvantage, the light source is somewhere, you need the connection. Advantage is if you have a huge laser, it doesn't matter because it's outside the body. If the laser gets hot, it's not the problem of the waveguide and the brain, it's somewhere else and you can put some water cooling around or something like that. But the challenge is how to connect that. We've seen that here with those mice. This was sold as freely behaving or freely moving animals, but again, this is something I would not like to have as human being, neither in myself nor in one of my children or my wife. You know, having something where you have the waveguide sticking out and you carry a light source with you around. That's probably not yet there where we like to have that. In that case here, as sketched out, we have a skull-mounted connector. That is something that is very established in animal experiments in neuroscience. So that's not a showstopper. You have those head-mounted connectors and you could live with that. That means at the very end we have a transcutaneous or percutaneous implantation. Part is outside, part is inside. That works in fundamental neuroscience research on animals. It's not that popular in humans with those connectors sticking through the skin and being an entry port for potential infections. So, again, that sketch, a bit similar to that what we had in the movie. So it could be either a channel through the membrane, so direct interaction, you shine light on that channel and the channel opens and either sodium ions flow in, then you have excitation of the nerve cells, or you have an influx of chloride ions here with halorhodopsin HR with yellow light, and then you remember that worm which froze due to inhibition. And then there are other things like opsins that are transmembrane proteins on the very right-hand side, there, where you have an indirect interaction. If you shine light on that, you have some release of CAMPs, so certain other things. And all of those, and this is an old slide, that means we have probably now 10 times as many opsins, come with a different need of wavelength, of energy, and temporal response. So we are between 10 milliseconds, switch on the light, switch off the light, so these are called the fast ones. And we have slower ones, which are more kind of step function. You switch something on, it takes a while, and then it modulates the whole behavior of the cells in the background. That means we have a variety of time frames here, and the fast excitation is much slower than the excitation with electrical current. We have there a single excitation, or let's say pulse width for electrical stimulation that are between 50 microseconds and 300 microseconds. And then we repeat them. Here the beauty is if you switch the light on, the channel opens and we have a repetition then of excitation. To bring that together in a nutshell, at the end, the light causes increased excitation or increased inhibition. The thing that went a little bit fast with to my thoughts and my imagination is the thing with the viral vector. So to get that thing working, the optogenetics, we have to create a virus, or we have to create a genetic sequence with the opsin and the promoter and a very specific part that couples to very specific cells. And we have to believe that this specificity is true. That means whenever we inject that vector, relatively unspecific. I mean, it's specific site-wise. You take a syringe and you poke your syringe somewhere in and you inject the fluid. That means if you do that in your arm muscle, the probability is higher to have higher concentration in your arm muscle, and then it diffuses somewhere. And if you put it in the brain, it goes into the brain. If you put it into the eye, it goes into the eye. If you put it into the cochlea in the inner ear, it should stay there. I have not seen yet any studies which report if those vectors really stay in place. So that's one thing where I'm a bit suspicious. Do they really stay in place? And there are already discussions about the efficacy. So how many % of the cells that I could target get transfected, and that varies from place to place and from species to species. And how long does it last? We all know from vaccinations, they last for a certain period, and then you're either good or not. And the prediction on that, how good your response on something like that, varies a lot between persons. And that would mean if you talk about a lifelong therapy, for example, the probability is very high if you transfer that experimental method into a clinical treatment that you need to re-inject that virus again and again over decades of a lifetime if it should work with the same efficacy. And that's more my topic here, is that interfacing part. How can we interface the nerve cells in a way that we can optically stimulate, and if we'd like to do that, electrical record. And what is that good for? So are there already some ideas? The ideas are manifold. So in psychiatric disorders, there are a lot of ideas how to treat that. Depression, anxiety, that should be obsessive compulsive disorder. There's an O missing. Spinal cord injury, hearing. There's a group in Göttingen doing translational research with Patrick Ruther's group here at IMTech on hearing light. So having a cochlear implant that works with LEDs instead of electrical stimulation. Vision, if you get blind, wouldn't it be nice to have injection into the retina and then you can see again without any implant? So cool stuff going on. The question is still about the side effects. Do we know something? Is this something that comes later? And is the claim true? Are we really more specific? And another claim is, electrical stimulation suffers from the fact that we have foreign body reaction and scarring around the implants. Does that matter if we have optical implants or not? So question, which light wave are we using? Infrared is probably fine. I personally doubt a little bit with yellow and blue that we don't see any attenuation or scattering if we have scarring around those implants. So that was here an overview from the internet. This is probably something you might read later on. So what pitfalls might we face? So one of all is that the delivery of light might be varying. So the effect might also vary then. And the question that still needs to be investigated, how much stress does optogenetic stimulation pose on the cells? are they still all right afterwards, or are they so stressed that they behave differently? Is that synchronization of activity? Same with electrical stimulation. It's not that asynchronous as the brain does, but really we fire and we activate a plethora of cells at the same time. Is this good? What about the target specifications? What about antidromic activation? That's always a huge question also in electrical stimulation. We have the synapses that guide the activity from one cell to the other, but what happens if it goes backwards? How does that modulate the whole network of cells and then the function at the very end? And what does that virus do inside the cell? Is it only the generation of ion channels, or does it interfere with other processes in the cell? Still things that I believe should be addressed on the long-term run. Which does not mean, well, we should not use that in experimental neuroscience. I'm a great fan of that, but I think we should put the bar at a realistic level if we promise new therapies. And it's always not fair if you suffer from a rare disease or from something that is really serious and you find those headlines in the newspapers, in the yellow press, that new therapy will come tomorrow because it worked once in a mouse. And this is something where I like to point your attention to. If you read that there is that attitude over the last years, which I love very much. If you look on new research papers, there's something that is called a graphical abstract. So you have a picture and 3 or 4 lines of description, if you should read the paper or not. And as a picture tells more than 1,000 words, and the abstract is limited to 200, so it's always awesome. And people cheat like crazy. You know, you see a human brain with a novel device, and saying, we tried out optogentics or electrical stimulation for whatever purpose. Said, oh, cool, human brain, description about the technology, and then you download the paper and you read through the paper, and it's on acute implantation in mice. So take care. The graphical abstract is a marketing instrument, not a scientific one. So it should be close to what you've done, but take care, especially if it comes to species. So that is that trick, you know. We can, after some weeks, we can all differentiate the sketch of a human brain from a mouse brain, right? And if we see a human brain, I always believe this is for humans. This is already in humans. But in many cases, it's just, let's say, an overview, say, well, in 20 years we might be there. So take care. So how does engineering now look like? And one of my favorite slides there is already is here, and that is the challenge of reproducibility in engineering. If you and I do something here at university, we have highly trained persons, PhD students, postdocs. The least trained person is a student assistant, which means you are in most cases more educated and trained than any production employee in a company. And then we have those PhD students, postdocs, who are generating a device, probably even 10 devices or 100 devices. That's good, but if you calculate that, you would be never able to sell that because your price is so high that nobody will buy it. And that will mean if you like to transfer that into product, that the costs have to drop. That means either splitting the processes up in simple tasks that a production person can do, or even automatizing everything that you press a button and come back after a week and your device is ready to go. But then the numbers come into play. If you do that for smartphone chips, there are millions of smartphones a year, that works well. But if you come to Foundry for integrated circuits and say, hey, I'm the most successful employer of the year for medical implants, I need 2,000 chips a year, they laugh at you. That's not worth for them starting a process, 2,000 per year, if you compete with 10 million a year. That means you run into some challenges there in supply chains, in manufacturing and reliability. And this is not our task as scientists, but we should be honest in communication. So, and know that there is a huge step between the proof of concept and the product on the market. And we have requirements on stability and reliability that go beyond the interface. So we need not only electrodes, we need an insulation and packages that goes beyond those connectors in super glue and epoxy resin. We need cables and connectors, either through the skin or under the skin, that we have a modular system that we can implant. That is not like implanting a spider web, but having something that you can connect and exchange if one part fails. It's the obvious on the biological reactions and the non that obvious human in the loop. We have doctors, nurses during implantation or application who are probably there with a completely different skill set than engineers have. And what is obvious for engineers in handling is not that obvious for surgeons and nurses in handling. And therefore, we have to have a lot of discussions, usability meetings to find out how should things look like. And at the very end, we have the patient who does not listen. I mean, I don't say that the medical part is listening to introductions, but the patient even listens less. And so the most famous failures in cardiac pacemakers were on the cables, because bored patients, while watching TV or talking, were trying to turn their implantable pulse generator under the skin when they were a bit bored. They played around and they turned it, and by that they broke the cables. One of my friends and colleagues, Greg Swanning, is currently here for 1 month internship from Australia, the University of Sydney, and he started his career in Cochlear. And his first failure case was to investigate why cochlear implants failed in 12-year-old boys in the rehab hospital. And that was a tough one, because he got the cochlear implants back, they were explanted, they failed, and the coil was broken. So the coil was no longer on the package. And to cut that long story short, that broke because during rehab, the boys found it really cool to run together down the staircase with an iron bar, and you can put your magnet on the iron bar and run down the staircase. That was exactly the correct height on that for those boys. And you can imagine that there was a vibration and the wire broke. The solution, and this is nothing you find in a design textbook, was to put another loop on the wire that you have a kind of lateral spring in that. If they do it afterwards, it's not a bending of the brake, but it's a spring movement, and then that it stays stable. I mean, this is something, if you are the responsible engineer there, you're getting crazy, right? And therefore I put it in here. So important to find out what do people do with that, and how to design that in the translational phase from the rats over the monkeys to the humans in daily life. And all the things that are valid, this is that very busy slide, all the things that are valid for electrical implants are valid for optical ones. It's the question on electrical shock. Are they safe? What is the connection technology? In LML experiments and later on in humans. How to stimulate? Do I want to have single nerve cell activation? Do I like to activate networks? That influences the design. Then all that stuff that I already told on the opsins and how much light do I need for which time frame, and all the foreign body reactions. I mean, just because it's an optical implant, it doesn't mean that the foreign body reaction changes. And that means we have ions, we have water, we have shift in pH, we have enzymes that like to eat up everything that doesn't belong there, and that all those things do not have to influence the functionality of the implant. And the question is that true or not? And the design with respect to the question where to place the light source. Do I put the laser diode directly on the implant? Do I put the laser diode into a package and then waveguides? How many laser diodes do I have to place there? What material do I take for a waveguide? And how do I couple the light in and out from one component into the other? Which materials do I take? And if you look in standard optical connectors, you find a lot of plastic glues. And if you put plastic glue into salt water, it tends to swell. And it tends to suck up all the humidity. So this is a good idea for a laser diode that you want to drive with 30 volts and have salt water around that. I wouldn't think so. So that means we need to take care how to encapsulate that, how to make interfaces from the technical side. And this is now just a wild rush through some things that we did and some things that other persons did. And we thought it could be good to summarize that all into kind of tutorial and wrote a very lengthy tutorial. We were supposed to write 10 pages in the first version, ended up with 120. And then we had to cut that down to make us and the editor of that journal happy. And we ended up, I think, with 30 pages or something like that. And if we have light sources, we need to couple out the light in a way that the light source is in a watertight package. That was our idea. And if we do so, why not integrating lenses in there? Then you can already focus the light and to guide it somewhere. That is easy to draw in PowerPoint or in whatever program. It's much harder to do that because you face a lot of challenges that the joining techniques are not that nice to your material because you need a lot of pressure and temperature, and then you have glass, and the glass has a different coefficient of expansion than your other materials. If you make feed-throughs, they're kind of holes into the package. That means whatever you do, you create a new hole to guide the electricity out and things like that. So there's a lot of knowledge there, but it should be always revisited with respect to the application. And we have different substrate materials, different light sources, lasers. We have integrated ones, so-called VCSELs, that is in semiconductor technology, very cool, very small, 300 by 300 micrometer foot size lasers. They unfortunately emit the light transversal in line with their footprint, and you normally want to have it orthogonal to that. So how to bring that around the corner. If you package those directly with some materials, they change their optical properties and you generate only heat and no longer light. So a lot of lessons you can learn with that. You have to see where to put hermetic packages around, so where to keep the water off, and where can you afford some water vapor around there. And if you combine that with recording electrodes, the question is where to place the laser diodes or the waveguides, where to place the recording electrodes, and are they close enough together? And do they really have to be close enough together? That's application specific. There are some persons who like to stimulate in one part of the brain and record in the other part of the brain, but you have to find that out. In that case, you do not need probes that can both. Yeah? In other places, you really have to be closely together. Yeah, and the question is, how does this stuff look like on the back end? Important, since the back end is not very sexy and therefore not always published in those papers where you look up all the latest news, and I have some figures for you here. So that is an overview, and I kindly ask you, if you dive into that, to look back before 2015 or something like that. Always gets a bit nervous when people claim this has been already done before and show you then a paper from 3 years ago, and you know there has been something even 20 years ago. So it's really good to look back to the beginning in certain cases. We have here in those tethered systems, that means those systems that have a connection through the skin. Either I start here on the right hand side, silicon, with integrated waveguides here and recording electrodes. Very cool with optical splitters, so you can split up the light. You can have it on polymers. This is one of our very early approaches where we learned a lot what we shouldn't do at all. And in both cases you see that the waveguides are relatively bulky with respect to the substrate, because you need to put the light through. This is here the waveguide, this is the substrate. There are approaches where you also integrate microfluidics to inject the opsin through the probe, but you know where it is. There are pros and cons to do it like that. And a very cool approach that, unfortunately, I did not invent that, but I find it really cool. Polina Anikiva from MIT is working on that. It's drawing fibers. That sounds really a bit boring, but the cool thing is you take a huge piece of material, like a polymer, you put in wires, like thick like my finger, you put in tubes, you put that all together in a kind of cylindrical block, and you need a huge machine. Huge means 3 stories high or 4 stories high. And then you heat it up and you use non-linear material sciences, and then with a multitude of the gravitation of Earth, you heat it up and you pull it down, and the material forgets all its laws, how it should behave, and stays together and gets thin. And this is here a diameter of 300 microns, where you have 4 or 5 wires in, you have a waveguide in, you have a microfluidic channel, and you have a cladding around. And if you do that in a really cool manner, that was what I've seen this year on a recent publication of Polina Anikiva, you can have hydrogels around. So the functional elements are relatively stiff, and all the rest is a kind of hard water. So that's really cool, but the technology is not that easy to understand and to make it reproducible due to that non-linearity in mechanical engineering and material science. And even more serious is the assembling technology at the back end. You have to fiddle out the electrical contacts, you have to fiddle out the fluidic channels and the waveguides and bring that all together. And this is still not really solved. But all that is still through the skin. Question, can we integrate that all, that we have a microsystem? Well, we would end in a telemetric backend. And if you look on that, you quite often see a lot of nice devices here with a lot of glowing light sources. Very cool stuff, either planar or on shanks. So where you have a shank design and then you turn it around, stick it in, or 3-dimensional stuff. My favorite one is that you have a Mozart, a music piece running in the background of a supplementary video of a publication, and like in a discotheque on the dance floor, those light sources are blinking in the right frequency. So very sophisticated technology. the question for me always is in that spot, okay, where is the piece which drives the electronics and the optics? And before I show you something like that, some physiological responses. So sometimes you need to do the obvious. For example, having an array here with a light stimulus in the middle, and you have to prove that you get more activity the more light you put in. I mean, that's, for me, it's obvious. If I have cells and they respond to light, the more light I have, the more cells see the photons and can react to that. But in that research on the interface, you have to show that, and that's shown here. If you have 1.24 milliwatt per square millimeter or 24.4, which is a factor of 20, you see a single response. There are underlying recording electrodes here. If you put more juice in, you get more activity. It's not very surprising, but it's following your hypothesis that you have. And using shank electrodes, one in blue, one in yellow, having transfected cells, you can record here single unit action potential, and one dot is one action potential. And then you have those post-stimulus time histograms. That means counting the number of action potentials over time. X-axis is, y-axis is the number of spikes here, x-axis is the time. So that works well, either exhibiting or inhibiting. Questions to be addressed there is, for example, the longevity of the waveguides and the coatings. And we took the standard coatings that people use in the clean room. So low temperature silicon oxide, low temperature silicon nitride, but you don't interfere too much with the temperature during deposition. Parylene C on silicon oxide, and we've seen that the coatings go away and the parylene C gets opaque. That's not good if you want to shine light through. We also see a change there. Well, we had silicon carbide. Silicon carbide on the left-hand side, that graph shows that the transmittance is really lousy. Means we have only about 30 to 40% in the visible light range, but it stays stable over time, which is not reversed. Silicon carbide left, SU8, one of the famous polymers in microsystems, stays stable and has a much better transmittance for the visible light. That's good. And it also works in vivo. So stimulating, the blue bars are the light stimuli, and the lower track is the recording. We see a slight drop in signal to noise ratio, which could be caused by foreign body reactions around the probes, that less light is getting through the scar around the probes. But it works well. Second challenge that we have with the system characterization: if we have light sources that generate light, this light generation comes with heat generation. And if you heat up your target tissue, then you destroy the proteins. That means you should stay below those famous numbers of 40 degrees C, probably even 39 to be on the very safe side. But if you switch those LEDs on, you see that you easily go above that. And therefore you need strategies. Either switching, we've seen those opsins are relatively slow, so if you switch fast, you have an on-off ratio, and you can keep the temperature on a lower level. And then my favorite one is the back end. Left-hand side is a standard one, and what you do not see in the red circle is the array. That means the array here consists out of 5 shanks, span over 2.5 millimeters. So shanks are probably less than 100 microns in diameter. And you have a back-end card on the size of a shoebox. The mouse has still the same size. That is not really something for freely behaving animals, you know, if you have a shoebox on the head of the animal. And this is not to bash my colleagues. I would have done it exactly the same way. But this is important to know. If you increase the complexity of a system by having 1,000 channels, and we have now claims in the US funding programs who want to go towards 1 million channels. The question is not how to make 1 million needles or 1 million contacts. The question for me is, how should I read them out? How should I couple in? Am I allowed to multiplex them in a certain manner? And do I really need 1 million? Or was the question, could you put 1 million in and I select the 5 best that I need? That's a different question. If you read out 1 million with 100 kilohertz or 5 with 10 kilohertz. Yeah, so that's very important to keep in mind when designing systems. I love the right-hand side system very much. That was, I think it's from Stanford. They took a very simple system and made it small. So these are small one-channel devices, but they are so small that that's a penny there. So they're really 3 millimeters long, 200 microns wide or something like that. So really small. You can implant them at certain spots, but I also would doubt that you can implant 1 million in the brain or something like that. So, but they're so small and they can work wirelessly, and with that you can investigate really freely moving mice in fancy environments and over time. That means over weeks or even over months. But taking the upper left-hand side and bringing that into a human implant would really be a very long way to understand what do I need, how to cook the electronics down, what kind of multiplexing would be adequate. And that brings me, oh, the slide number is vanished, completely vanished, or just hidden somewhere. No, it's complete. It's down there. So the box was to show it. Should have checked that out. So 24. And that means we always have to check what we really need in the design phase. How many optical channels do I need? Where should they be integrated? Should they be integrated? That's probably one of the most important questions. If I want to see how localized optical stimulation in the midbrain influence network activity on the cortex, I can probably take a surface array for cortical network recording and combine that with waveguides to stick in deeper areas of the brain. That's nice for fundamental research. Yeah, and then you don't need to have that device that can everything. I have my history with those devices and learned a lot. We did that one, we published that 2013, we started about 2010 when that Method of the Year paper was out, and we thought it would be nice to have electrical recording, optical stimulation, and in those days the question always was, do we transfect the right area of the brain or not? And therefore we thought we should integrate some microfluidics, that we inject the opsins really on that site where they are. And that took a while, and the front end looked quite beautiful here. The channel output for the microfluidics, the waveguide here, the electrodes to have different spots, left-hand side SEM, electron micrograph, right-hand side the light microscope, and the upper one is the nightmare. That was the connector. So the front end is here. Well, we need an optical connector, and this is one of the standard ones where you can have that winding to put a glass fiber on top and close it. The glass fiber is thicker in diameter. That will mean the fluidic connection must be apart from that, otherwise you cannot connect the fluidic connection to that if it interferes with the plug from your waveguide. So the microfluidic connection went around the corner, and then they said, and by the way, we use those standard Omnetics connectors, 16 channels, and we use them always. And you can imagine implantation procedure was the killer criterion, and a lot of those devices failed just by implantation because one connector went somewhere and tore the whole thing apart. If you look from nowadays to that, I would completely agree that this was one of the most stupid designs we ever did. But in those days we thought, this could be a way how to handle that. Later on, we thought, on one hand side, if we have light sources directly on a surface array, we can integrate them into packages and have transparent lids with integrated beam-forming elements, so lenses. That worked out well, but was more complex than we thought. And we thought we can use old-style semiconductor technology to make electrical feed-throughs without metal-silicon connections. By using that thermal migration effects that I introduced to you during the Utah array, that you put some metal on one side, then you heat it up, you just need the gradient, and then we found out that there is nearly no oven around that is able to do that gradient to let those channels grow. Well, it somehow worked. What I thought is easier, but turned out to be even more complex, is to couple a glass fiber to a glass, let's say a waveguide that goes through a package wall which is transparent. Many approaches use polymers there. And using polymers as material through a hermetic package is like putting a wick into there. These polymers suck water like crazy. There is no water in the package, there's all water outside, and it doesn't take long, 48 hours, and the whole package is flooded. I mean, that's nice if you want to prove that theory is valid, but it doesn't help if you want to have working systems there. And we thought we can borrow that technology that you heat up glass and press it into forms. And the company promised that this works. It didn't work at all. But we found out some parameters how to combine things in a way that you get, that you have a combination of standard MEMS technologies, standard packaging technologies, and waveguides. But this is also something where you should think twice if you really need such a system for chronic implantation, because it gets really, really expensive. And that means it must be a good reason to use that, otherwise you'll never get that in neuroscience, and it could be hard to transfer that into clinical applications. What was a bit surprising for us, well, it's not that surprising if you look into material science, but what turned out to be a promising material was silicone rubber. All the others took materials that are more sophisticated, so having really tiny little silicon oxide waveguides on silicon probes, having SU8 probes, having glass probes, whatever. That's fine if you use technology where these materials are kind of merit to the technology, but if you use polymers, we thought, why not using other polymers? And it turned out that silicone rubber is very stable. The downside is that the process technology needs to be very precise because every tiny little air bubble in there or dirt particle scatters the light and you don't get anything out. But at the very end, it turned out that this works well and we can transmit amounts of light with respect to energy density that are available as in standard equipment, and you can excite nerve cells that have been transfected before. Still wants to get published, those data. The downside is you need a large size. The beauty is silicone rubber is very soft and bendable. That means for certain applications it might fit, for others you would need smaller things that come then with higher stiffness. And it works still after a year in hot water, so accelerated lifetime with the idea if you heat up something then diffusion goes faster, and if you have diffusion related processes you can predict the lifetime in a shorter period. I mean, no company would put their devices for 30 years in a saltwater tank and comes up after 30 years. Now we have proven that they work, now we can go to sell them. No, then they're completely outdated. You have to cook that down and find some measures that you can probably perform those tests in 1 or 2 years before you want to bring something to the market. Just as a side note here, what turned out to be as challenging as the whole optics integration is the integration of feed-throughs. And this is completely underestimated, and we're currently trying to publish those results. The showstopper is in most cases the connector. You take so much care of your probe design, and then you put it to a connector, and the signals, they don't get short-circuit, but they experience crosstalk on the connector level because the resistance between those lines is least there. And if you have signal on one line and the other line is there with a low insulation resistance, you might have a coupling from the active line into a passive line. And if this is an action potential, it has high frequency components, and you can detect action potentials where they shouldn't be. That's really devastating if it comes to network activity investigations, where you want to find out which parts of a network contributes to what, and this is mainly that destruction of the signal mainly on the connector level. The funny thing is, if you know that, if you know the characteristics of that, you can use algorithms to eliminate that at the very end and to clear your data. That's very cool. Sharif did his PhD work on ideas how to make those connections under the boundary conditions that they should not short-circuit each other. And this is important to keep in mind if we talk or if we read about 1 million channels that are recorded somewhere and they need to get connected. Are they really electrical insulated on the connector level? And we had here an approach where we used silicone rubber. That was completely crazy because silicone rubber is forbidden in cleanroom processes since it screws up all the cleanroom processes, and so we needed to have that in a way that we can use it after things came out of the clean room, and had a kind of dry insulator in between the package with a connector and the array. And then we insulate those contacts by a layer of silicone rubber. Comes with a lot of challenges that I don't like to comment on here, but the take-home message is it's really good. So we have, if we need that, a factor of 10 in comparison to commercial connectors. And that might be worth following that approach if it's needed in the application. If people say, I don't care if I have 20% of crosstalk, then it's hard to negotiate. But if you want to have less than 5% crosstalk, this could be a solution. And with that, I'm close to the end. Since I still some minutes, probably I can go back to Sharif's thing to show you something, because that is a second challenge in interconnection. You have to follow three properties. One is the electrical connection. You want to connect all your contacts. That's obvious for a connection, right? It should be mechanical robust. If you connect something, it should not fall apart. And the third thing is adjacent channels should be insulated from each other. And it would be nice if you could, at least in experimental settings, if you don't go into mass production, to visually inspect your contact points to get an idea, is there something good or not? And one of the challenges is with silicone rubber, that silicone rubber does not like to stick to most of the materials we use in the clean room when it comes to polymers. So, does not like to stick that much to Parylene C. It does like to stick to silicon oxide. That means silicon oxide is always present on silicon. That works. It does not stick at all to polyimide. If you want to make it stick, you need to have some interlayers in there that keep it together, and this is what we did. So we did some silicon carbide on top of polyimide to have then polyimide, silicon carbide, the carbons bind to the carbons in the polymer, and then we put silicon dioxide on top of that, which bonds to the silicon carbide, and then you have oxygen groups that love to bind to the silicone rubber. So you have to play some tricks there. And one of the meanest things was there that silicone rubber is not really compatible in thickness with microsystems. So we have 10, 20 microsystems height, which is not that much for a normal person, but a nightmare for a microsystems engineer. And people thought we are crazy, but we love that approach. We had the idea we create springs. If you have a spring, like a spiral, you can press the spiral down through a hole in the silicone rubber to the substrate underneath, and it doesn't matter if this is 5 or 10 microns higher or deeper, so you can allow certain thickness variability. And if you shape the front part properly, your bonding machine connects your spring via deformation in the bond over the bond with the substrate. This is a kind of a bit misaligned riveting. And funnily, that worked out well, and we could rivet all those springs to the substrate, and then we poured silicone rubber on top, and all the holes were filled with silicone rubber. That means we had a good insulation to the outside, good insulation to the neighboring part, and a good insulation resistance, surviving temperature shock, and vibration. And I think that's, and then it turned out that people said, oh, we don't need so many contacts. 230 is too much for us. Can you do 4? Okay, we can, but probably without that technology then. Good. So that's now at the end. Guess you get at least an idea how microsystems and optogenetics come together. That was not about the beauties of optogenetics to interrogate neural circuitry, but I wanted to have that other point of view here on optogenetics a little bit to get you an insight. What are the challenges from the engineering side to make things work? And to summarize that, so yes, we can genetically modify nerve cells, and then we get that cell-specific behavior. That means shining light on the cells. We have probably only the interneurons that react, even though all the other cells are grouped around with that genetical modifications. There are a lot of optogenetic tools around. Some are more do-it-yourself-like, others are very sophisticated here, and they serve this method. I would still say that most of the applications are on a fundamental level. More and more are now going into clinical trials to see if it works into humans. But we have to see what comes out. Probably I'm so pessimistic or let's call it realistic because I'm growing old and have seen several developments. There was a huge fuss on stem cells, for example. And beautiful ideas. Stem cells to get progenitor cells injected in the retina if people suffering from retinitis pigmentosa to make them see again. Stem cells in Parkinson's patients to inject them in a substantia nigra that they regrow and secrete dopamine. So stem cells to get injected into the heart after heart attack. And some things worked and others didn't. One of the first studies was stopped in the US because people got more cancer. Yeah, so sometimes cells don't stay where they are supposed to stay. They travel around the body and they do what they're programmed to. They grow. And if you grow in an undefined manner and an undefined place, we call that cancer. Right? So we have to check that out. And that is the reason I still say we should check it out where the opsins stay and how long they stay and what does happen if I re-inject things. I mean, the vectors, are they really eaten up by the immune system or are they just silent? And what happens if I come over a certain threshold of concentration? That could be all fine, but we should be aware that we should check up side effects and hidden mechanisms there. Yeah, as said, that hearing light thing is going on. Guys, collaboration between Tobias Moser in Göttingen and the Fraunhofer Institute in Dresden, they have already funded a company to fiddle together all the options they need, and Patrick Ruther is doing here the assembly of the optical probes for cochlear stimulation. And I'm really keen to see how far they get and if they're really able to have a good outcome there. And I would love to see that because I think if cochlear implants could be improved by certain technologies, that would be a huge step.
What would be the improvement actually?
Good question. I thought I would get around that question. No, so why should we stimulate the cochlea optically? We have the ganglion cells, well, we have hair cells, if we hear normally, in the cochlea. And if they die, we still have the ganglion cells. And the ganglion cells are, let's say, on the inner side of the spiral. If you implant an electrode array, it tends to lie on the outer side of the spiral. You can try that out. Make a conus and try to wrap something around. And that would mean, and if people found out how to place the electrodes that they're really facing towards the inner side. First, implants at rings and spread the current everywhere. Now you have already those contacts to the inner side, but they are at a certain distance there, and that distance would cause overlapping of the electrical fields of adjacent electrodes.
Okay,
and that means that the spectral resolution that comes with a distance between two adjacent stimulation sites is somehow limited just by the physiology of the electrical field. You can play some tricks, and that is already done with the speech processors, that you say, I do not only rely on the spatial arrangement of the frequencies over the cochlea, but also on a kind of higher order mechanisms where the nervous system detects the zero crossings of the frequencies. And that means if you predict to stimulate in a manner that you code the zero crossings of different frequencies, you can get better in the fine structure than the spatial resolution. So you predict a higher order of sine wave just by stimulating kind of zero crossings. And the idea with the light is that the cross sensitivity or the crosstalk between adjacent light sources is better than the electrical stimulation.
So you could basically get a higher resolution.
Yeah. I'm still want to see the results. That could work well, and there are first results that show that it works well, but we do not know what time does, right? If you get a bit more opaque, if there's certain proteins there, they might scatter the whole thing, and the question is if you can maintain that resolution then over, let's say, 20 years. And this is still unknown, but that's the basic idea. And they say if we make good opsins, whatever good opsins means, we can reach that goal.
But is it limited by the opsins and not by the light?
In both. You need selective opsins and then selective microphotodiodes. I could imagine, I said opsins because I already have in mind, probably you can code adjacent frequency bands in different colors.
Okay, that would be amazing.
You know, like a color TV,
Yeah.
and then you can code that and overcome even the optical scattering there.
That would be a genetic question then, because you need other promoters that act. Sounds confusing.
Yeah, that was just my idea if somebody would ask me for a blue sky research project. I think there's a long way to go, a lot of exciting things, and I mean, you always get new knowledge over pitfalls. So there will be success stories, there will be failures. You can draw conclusions out of the failures and try the next success story out of that. So that's research. Yeah. Good, and we have to solve a lot of problems so you don't run out of work in your careers. And I hope I could show you it's something completely new, but the fundamental question when it comes to implant development is similar. So it's all about stability, reliability, longevity, crosstalk, probably different crosstalk, but all the fundamental questions are still valid in those devices. Yeah, and with that, I like to recommend our own articles, a little bit the marketing thing here. If you dive into that, we learned a lot while writing those articles. That's always something good. Since one has been published in 16 and the other in 18, there are new developments since then, but the general questions are still the same. so behind the whole storylines. And with that, I'm at the very end. Like to thank you for still being here, and want to remind you that I'll invite you to another lecture or another session next Monday, where I'd like to go with you through the general concept of the exam and some hints what I think could be a clever strategy how to prepare that. Yeah?
If I remind correct, in the lecture notes there's one lecture about biocompatibility.
We already had that.
Yeah.
So we switched that, the cardiac pacemaker sensors and the biocompatibility. While I was traveling, Ali took that 12 biocompatibility, and we thought we keep the numbering as it is, that you can assign that better to the lecture notes as PDF.
Okay, yeah.
So we had that weeks ago. Just as a reminder, that was switched. I think it was with sensors and cardiac pacemakers or something like that, which came later on. I screwed up the recording of the telemetry, so there will be an upload of an old one. Technology, words are a bit different, but content is the same.
What was last Wednesday for instrumentation?
I sent you all an email 2 days on Monday, and the system, I'm sorry about that, I have to apologize. I sent you on Monday an email that Wednesday does not take place, and the system said me, I sent out that it sent out 50 emails.
I think like 40 people were here.
Yeah.
Very sorry about that. I took the Ilya stuff, and there is something like send email to all participants, and I got a confirmation email that the email has been sent out, and therefore I thought you are all informed, and I've seen that you sent me an email. I have to apologize. I thought you were all informed on that. We had a TÜV audit 3 days in a row for an ISO certification, and the TÜV was interrogating, and the boss should be there with that. And I thought you got that. So apologies. We are still in time with a lecture on biomedical instrumentation that we can cover everything that you need.
So we do it again? Or do we just take the online recording?
No, you get the safety, if you like, on Wednesday.
Okay.
And I hurry up a little bit to give you 10 minutes ideas how an oral exam will be. Thank you.